So the process can be schematically put out like this. This is a very simplified process, but basically you have some sort of gene source. There's a plant that, or a bacterium that is resistant to a specific type of bug. And so we look through, and this is why we sequence genomes. We find the gene that controls why the plant likes to avoid the bug. We choose that gene and we cut it out and we make a whole bunch of copies of it. Then we find a host organism and we use plasmids. Plasmids are circular rings of DNA. They're basically the chromosomes for bacteria. They're just more simple than plants and animals and their sets of chromosomes, and so plasmids are easier to deal with. And what happens is we put the desired gene into the host plasmid. We cut out a section of the plasmid using enzymatic cleavage, and then we put the new gene into the hole that got left. That's a process called insertion. So in insertion, we put new genes into old plasmids, and we form what's called an interspecies vector, right? This is no longer a genome, it's no longer a plasmid for this bacteria, it's interspecies. It's got information from two different things. And then we insert that plasmid into a host cell. Right? That interspecies vector is transferred into a host cell, and there's a genome in there telling the host cell to do things. And that host cell is saying, make these new proteins, and these new proteins are resistant to that bug. And so now the host cell, just like the source, is able to perform a function that we wanted it to be able to do. This is how we genetically modify crops. We take a host plasmid, we put some sort of, say, drought-resistant gene inside the plasmid. We put the plasmid inside, we infect the plant with the guest gene, and the guest gene makes the drought-resistant proteins for the new cell. That's what happens in genetic engineering. We're putting new instructions into a cell. And you can imagine that there are benefits to this. One of the benefits to genetic engineering is that you get better synthetic processes. If you're diabetic and you take insulin, it used to be, up until about the 80s, that you took insulin that came from cows or pigs. It was close enough and it was okay. Now they can use E. coli bacteria to synthesize human insulin in much larger quantities. They don't have to use the almost there pig and cow insulin. They can make real human insulin from bacteria. You can get more environmentally friendly processes out of this. So instead of using a lot of really expensive or really nasty chemicals, you can do things in a more environmentally friendly way. So for example, this little disc here is plastic made from algae. They can stress algae genes to produce and overexpress a certain type of protein that causes algae to make monomers essentially, and those monomers can be turned into feedstock polymers for new plastics. This is better than, say, making the polyvinyl chloride monomer. So the PVC monomer requires chlorine gas, and it's not a very nice way or an environmentally friendly way to make things. You can get stronger crops and improve nutrition. So right here we've got some peanut plants, and these are the non-modified peanut plants, and you can see that the bugs have been having a field day with them. On the other hand, if you insert a gene into the peanut plants that causes it to make proteins that make the plant resistant to the bug, you can get much healthier plant, same season, but much more resistant. You could improve nutrition too. There are rice plants, for example, that have had extra vitamin A producing proteins inserted into them so that people get a better source of vitamin A throughout. Also, new ways to fight disease. For example, you may have seen in the news what are called knockout mice. Knockout mice are simply mice that are missing a specific set of genes. And that way scientists can work on a knockout mouse and they can try to figure out, is that the gene that causes a particular disease or is it not? If I take that gene out and I treat the mouse in a certain way, what will happen? It allows us to learn about disease. And in fact, we can even make drugs now. Just within the last couple of years, the FDA has approved what's called Atrin, which is a drug that treats people that have a disease called antithrombin. And antithrombin used to be that they'd have to take human blood plasma and they'd have to isolate the antithrombin and then give it to people. Now they can actually modify the DNA of a goat to express that particular gene that makes antithrombin. And then those kids simply grow up and they milk the kids, and that goat milk has the antithrombin protein that people need to treat that particular disease. So it's sometimes called pharming, PHARMING, where they're actually using animals to produce a vaccine or to produce a drug. Now, I want to stress that antithrombin is the only drug on the market right now. This is not a common thing, but it's sort of akin to putting antibiotics in chicken feed supplies so that your chickens are expressing antibiotics. The idea is to make your food sort of a one-stop shop for all sorts of different things. Of course, that leads to a lot of concerns, right? Is there a butterfly effect? If I modify this plant, will something down the line go wrong later? It threatens the natural balance. So for example, you could imagine that if I modify this corn crop, then all of a sudden I've stacked the deck in favor of the corn crop. Will I cause that corn crop to take over the area? Did I need those weeds? Did I need those bugs? What about evolution of the attackers? So those bugs aren't just going to get static. Could those bugs evolve to get past the bug-resistant toxin that the plant produces? This is a complex and expensive process also. You can imagine that, for example, in the last slide when I was talking about fighting disease, or if you think about gene therapy or cancer drugs or things like that, the idea is to make a designer drug that works for each person individually as they need it. That's a complex and expensive process. There are 7 billion people on the planet, and every one of them is unique, and so you have to think about and take that into account when you're synthesizing those particular drugs. And also the really big concern about genetic engineering, right? If I make my food a one-stop shop for vaccines or antibiotics or better molecules, have I created a genetically modified organism, a GMO, that could turn out to hurt me along the road? What are the ethics of making that plant? Who owns the plant? Is it the engineering firm that made the plant that owns it and nobody else can grow it? Actually, that happens a lot with genetically modified corn. So, for example, a lot of the genetically modified seeds have to be sprayed with a certain chemical to even grow in the first place. What happens when you buy the seed from that corporation and you have to buy the spray, and all of the world's food down the line could possibly be controlled by one corporation? Could that be a problem? Absolutely. That's a moral and an ethical issue. Cloning is a type of genetic engineering, and you may have heard of Dolly the sheep, right? What are the moral issues with cloning a human being? Why would we want to do it? Well, maybe we could harvest organs for organ transplants, or we wouldn't have blood transfusion problems anymore. But are those people? Are those not? What is the deal if they're cloned? What about, for example, they took the Vacanti mouse, so Vacanti is just the name of the scientist who was involved with the team, and they programmed this mouse to grow a human ear, or more accurately, grow the cartilage framework for a human ear, not the actual functions of hearing, on its back. Is that cruelty to animals? Is it safe? Is it a good idea? There are all sorts of concerns about genetic engineering that have to be hashed out. Science can't answer those questions about policy and whether we should do it. It just tells us how to do it. And what do we need to think about? These are important concerns for the future. Don't just know that because we can do it, we should. That's not the way it works.